morning, students. We are just continuing our class in specifically regarding this herbal drug, that is traditional preparations, and we are just continuing the stability study what we have seen. This is our third class. We have already finished our two classes, and this is our third class and about the stability study. As I told in the last class, we had this a chapter in our herbal drug technology itself, and which we had gone through about the various even we compared with the patent regulations and how to get the patents, and then we had a small chapter in that, and there we have explained little more little deep some points, and here some points, so you have to compare and make yourself to clear for your examination. Understand? Can you follow? That's why there is the aim, and I have given some of the details here also. Where this is the third class, analytical methods. That is, after we want to where we stopped in the last classes. Actually, we stopped with our various benefits of this St. John's Wood's flower, which is used for the premenstrual syndromes and also for menopause, and it has antiviral drugs. This is about valerian. Valerian is also a flavoring agent. It is a precursor for various products of flavoring reagents, and this is also we discussed in the last class. Now we are going to see about the analytical method of determination or determining the stability of herbal products. The analysis of herbal preparation is mostly done by modern chromatographic techniques or spectrophotometric techniques like HPLC, gas chromatography. HPLC and gas chromatography, both they used to have a discussion, and they used to make it a confirmation by means of the compound, and then TLC we are mixing up, and HPTLC, and also we used to do quantitative estimation by means of UV visible spectroscopy, and or a combination of HPLC and GC, and even MS mass spectroscopy, and nuclear magnetic spectroscopy can be used for structural elucidation, and method can be used for identifying the purity test as well as the detection of single component for the assay. So here now we have to know about a point of shelf life or expiry date. So there is some difference. I was telling what is the difference in shelf life and expiry date. A slight difference is there. That is also I have denoted here. So I hope it is necessary for you to know what is a small difference, as we have discussed in the last 2-3 classes before. We discussed about in the research methodology. We came to know about what is the difference between reference and the bibliography when you are giving a dissertation submission. So that is how here also we should know that what is the difference between a shelf life and an expiry date. Both are interrelated and very close form. Only a slight change is their difference. That's all. An expiry date is defined as the time up to which the herbal product will remain stable when stored under recommended storage conditions. Understand? An expiry date is defined as the time up to which 2 years, 3 years or 1 year, time up to which the herbal product will remain stable. It doesn't deteriorate, it doesn't get spoiled, it doesn't change its color, it doesn't lose its potency. So these all stability studies, when you complete, there you are telling that it is an expiry date. So expiration is defined as the time up to which the herbal product will remain stable when stored under recommended storage condition. This word we are using. When stored under recommended storage condition. What do you mean by this recommended storage condition? The manufacturer will give the recommended storage condition, in which humidity, in which temperature, or it should be refrigerated or freezed, or it should be kept in the dark, away from the dark, or it should be in a dark place, or it should be kept away from the light. These all things should be given instruction by the manufacturer after particular study made in the stability of the product which they have manufactured. And if you follow, that is the date. That's the date which we are telling. That is the product time up to which the herbal product will remain stable when stored under recommended storage condition. Thus, an expiry date is the date beyond which it is predicted that the herbal product may no longer retain fitness for use. It cannot be used. That's the date which they are giving. That is called expiry date. You might have seen even in all the drugs, even in the pharmaceutical preparations. So we are just mentioning herbal drugs, but the same thing is followed for all the drugs. Understand? Can you follow? Even it can be applied for all the other drug for stability studies. If the herbal product is not stored in accordance with what is the instruction given with the manufacturer instruction, then the product may be expected to degrade or decompose or deteriorate or lose its potency and to degrade more rapidly. Understand? Expected to degrade more rapidly. Shelf life is a time during which the product, if stored appropriately as per the manufacturer's instruction, will retain fitness of use more than 90% of the label claim of potency. So we used many words: potency, deterioration, color change, and losing its potency especially, and there is, for example, no longer the fitness of the product for the use by the patients. All this will retain until the expiry date, and you can say it is also shelf life. Then I thought, what is the difference between shelf life and expiry date? We are telling the same, interlinked. Yes, they are interlinked. You can see here the difference between shelf life or expiry date. Shelf life is generally related to drugs quality over a specific period of time, whereas expiry date relates to both quality and safety. Quality and safety. Expiry date, that is general shelf life. Shelf life can be given for product or anyone. We can say shelf life. Shelf life is a quality, relates to the drug quality for a specific period of time. But when you talk about expiry date, mentioned in the label, it states that apart from that, over that date, it is not to be used. It is not safe to use. So it is mentioned for both quality and safety of a medication at a specific point in time. That's all. Only it says an expiry date means after the date which is mentioned in the label, the product is not allowed to keep in the premises of pharmacy, or it is not for the cell, or it not for the dispensing area. It should be taken away from there. That's why you might have seen in all pharmacies they used to discount the expiry products, and they will not keep. They are not allowed to keep in the premises. They should be kept in separate place, and properly they might have been denoted that these are expiry product for destroying purpose. We have to keep labeled, and it should be removed from the premises. And now we are coming to the protocol for stability testing. Protocol for stability testing, I just made of 6 points to remember, like this, colored, and to show you why. Because students can remember at least the points and they can go for. You cannot read all the theory paragraphs and pages together. If you read, it will be difficult. Make hints and make yourself comfortable to write the exam in point by point. As I told, how you present your paper, how you present in the paper makes the evaluator to give you, because they spend very, very little time. Very, very minimum time. They don't have time to go inside the reading of all your matter what you have written here, written in your examination paper. So just make it clear how to, and no one who have taken quality control and of herbal drugs or standardization, quality control and standardization of herbal drugs as an elective subject, no one should come again for writing. No one. All should clear. All should, you should go with a good certificate for your 8th sum with very happily with your project work and other subject. Not only I am telling about my subject, you should, it is your responsibility. It is responsibility for the semester student who is studying now in 8th semester. You have lost lot of things. You have lost lot of things in your life. You have lost more than 1.5 years in your life due to this pandemic. So you just imagine that is this loss. If you want to correct it, you have to clear everything in this semester without any type of hindrance. You should, that is your responsibility. So try to, you see, I am just trying my hard. I'm also somewhat something lazy. Sometimes I am not thinking about my student. I'm just leaving the things and I'm going away from the track. But I thought, yeah, it's my responsibility even to give you a very good take the information about my subject, which will helpful for them to write something, and they should clear it. You should get a job. You should make your family happy. So please, you have this thinking and make it clear about your this semester. Don't take it lightly. Understand? You have lost many things, so you have to gain it back by means of your presentation in your examination. How you can make your family happy. Once you make your family happy, they are more happier than anything, because they invested on you and they expected from you the only result. They did not expect anything else. Come in the top, do the maximum. So now the protocol for stability testing. Sample planning is very important. We have discussed about the sampling also. Little more I have discussed in the initial classes. I have shown most of the videos and all these things. But anyhow, that is all something to improve your knowledge and basic knowledge which you have. It will be helpful for you to go for your examination. And it will be helpful for you in your future, in your interviews and in many places where you go for your work. And you see batches. Protocol batches. How many batches? How many samples? How to select a batch? And if you have 3-4 batches, if you have 10 batches, how many samples you have to take? Or you have to take only the first batch or the second batch? Not like that. You have to take every batches. How many samples you have to take? All these things today we are discussing in this class. Just revising. Sampling already we have come across in the first class itself. So here also protocol for stability testing also, from where you have to take sample. how many samples you have to take, what for you have to do, what type of formulation, what type of stability test you are going to perform. These all things have a parameter over it. And containers and closures. What is the protocol stability testing? Containers and closures, they are also having an impact in this. So protocol of stability comes to the batches, containers and closures, and the orientation of the storage, orientation of the storage containers, orientation means of the product which is stored in the containers have some orientation, leaching, have some reaction with the containers, materials, and orientation of the storage conditions. And then number 5, you can see frequency of testing. Frequency of testing. How long? 3 months, 6 months, 12 months. These all already we have discussed. So frequency of testing also a protocol for planning of a testing of stability testing for herbal drugs or herbal preparations. Testing parameters also comes under in this. There are many parameters which is coming under the testing. They also come under the protocol. Where to test, when to test, and how to test. All these things will come. So you see, this is what we have to see. There is batches, sampling, planning, containers and closures, the orientation of the storage of containers, and the frequency of testing and test parameters. Let's go in detail about these topics which I have given you in protocol of stability testing. First you have to remember again, protocol for stability testing, batches. We are going to discuss about batches. We are going to discuss about the containers and closures. We are going to discuss about the orientation of the storage of containers. We are going to discuss about the frequency of testing. We are going to discuss about the sample plan. And finally, we will come to the discussion about the test parameters. What are the various parameters which we are coming across? So you just now we can protocol of testing. First one, a well-designed stability protocol should be contain the following things. Number 1, which you have seen in the picture, batches we are taking as first one explanation. Stability studies at developmental stages are generally carried out on a single batch, while studies intended for registration of new drug products or unstable established products are done on first 3 production batches. If it is a new one and if it is an unstable established product, it's a new one for the market, then you have to take first 3 production batches should take in consideration for your stability testing. This is what the protocol, for you to go for a stability testing for any type of herbal products. While for stable and well established batches, while for stable and well established batches, even 2 are allowed. There are 3, well established and stable, 2 are allowed. The selection of batches should constitute a random sampling method, from the population of pilot or production batches. So pilot, what do you mean by pilot? Pilot means we may be pilot. I'm not telling about the pilot who drives the aircraft. This is pilot sampling. Research purpose we are not going to do in a large scale. So maybe a pilot sample research purpose, or maybe already marketing for already you finished all the phases of research study and it is going to produce for the market purpose. In that case, possible for you to take random sampling. What do you mean by random sampling? Random sampling means you should take random. It may be timing of the production time. morning, afternoon, evening, or it may be from the batches which is kept, or it may be the containers which you have kept, or it may be a heap which you have kept from the top, middle. So random samples you have to take, not from a particular one batch or particular one time, the production outcome, particular one sack, a sample taking, or particular from the top or in the bottom of the sample of the pack. No, it's not at all allowed. You have to take a random sampling, then only you can have an average of stability study. The number 2, containers and closures. Containers and closures. So the test is done on the product in immediate containers and closures proposed for marketing. So containers may be proposed, there may be plastic containers, metal containers, tablet means blister packing, aluminium packing, bottle packing. So everything should be tested. So the packing material include aluminium strip packs, blister packs, HDPE bottles, etc. So just I have taken this HDPE means what is HDPE? Bottles with high density polyethylene bottle. High density polyethylene bottles like this. Nowadays it is used for liquid jars. Larger jars are also available. Smaller jars are there. Square type, round cylindrical types are also available in the market. So aluminium strip pack, blister packs and HDPE bottles are, you have to check. You have to analyze the second one for the stability study. This is aluminium foil which is used for aluminium type of tablets, how they are in aluminium foils. They are strip packing. We used to say aluminium strip packing. This is blister packing. You can pack the soft gelatin capsules and even tablets. Nowadays most of them are using blister packing, blisters. The plastic, the polyethylene sheets are made into blisters and the product will be held over it, and above that there will be aluminium label or a sealed, what we have shown in the blister packing. Blisters are formed in the hard plastic of polyethylene. Plastic sheets will be made into depressions, made into this, that is a cavity, or you can say it is made of molding, which according to the requirement of the tablet capsules, which can be even sealed by means of keeping a sheet of aluminium foil. Some of them even they are using paper mixed. Polythene can also be used, cellulose with polyethylene sheets are also can be used for blister packing. This may also include secondary packing, but not shippers. Shippers means actually they are mentioning about the shipping. We are taking some cartons, cardboard boxes. But cardboard boxes also, when I was getting training in a manufacturing units, they put me for a 3 months training everywhere in all departments. In that even I went for testing for the containers and closures. In there, they have various testers even for cardboard boxes, cartons. What? Because they're busting quality, thickness of the cardboard, thickness of the material which they have used. So that test they will pass, then only they will allow the product to enter into the pharmaceutical industry. So there are many tests even for quality control test for even packing materials. Even when you keep a rubber cork for a vial, an aluminium seal for an eye drops, or a cotton pack or a bottle which is coming for eye drops, ear drops, they all will be checked. Thickness of the bottle, quality of the bottle, and whether the bottle is made up of proper borosilicate glass, what is the silica quality. All these things will be analyzed in the container closures quality control department. So that is what we are just discussing now for stability. Stability depends upon the containers and closures where you are keeping your product. So the product may be leached through the containers and it can have incompatibility with the containers. It can react with with the containers. So this all comes under the stability study. The product in all different types of containers closures, whether meant for distribution or for physician and a promotional sample, or should be separately selected. You know very well, every factories, every companies, they will be having sample for physician sample not to be sold. And also they will be having some promotional samples for even for the pharmacist who is dealing with various distribution. And also that these samples, all samples will be separately, they should be analyzed for their stability study. Number 3, that is C, that means orientation of storage of containers. Samples of the solution, dispersed system, like solution, dispersed system means we talk about not only the liquid preparation like emulsion, suspension, even you can see semi-solid preparation, ointments, creams. There also you can see there is 2 immiscible substances will be made to one to disperse in other. As we know the definition previously, you know very clearly about the emulsion. I think 2 immiscible liquids, one is dispersed in other by means of using emulsifying agent. Suspension, that is one is solid which is insoluble in the insoluble. So we made it into small particles which can be dispersed in, and viscosity can be increased and it can be suspended, or it can be using by reducing interfacial tension also it can be suspended. So that is, there are many flocculated, deflocculated. You might have studied more about the suspension and emulsion in your pharmaceutics. So there you are telling this liquid preparation may interact with the containers, and so dispersed system of semi-solid drug products. For stability study must be kept upright and positioned either inverted. You can keep inverted in the bottles or slide by, or on a side allowed to full interaction with the, you can keep it side by and keep it such that the solution will touch the containers of the product which container is in the closures, so that you can find this orientation. The liquid can up to, there is some larger bottles, how it is moving in the bottles, and these all things comes to the containers touching the solution with and having the connection or not connection with the contact with the containers can be observed. This orientation helps to determine whether the contact between drug product or the solvent and the closures result in extraction of chemical substance, whether the body of the containers and closures are absorbing the main constituents, reducing the potency of the constituents, or reducing the amount of the substance which is present in the product. So these all things will give the chemical substances can react, or absorption of the product component into the closures and containers, and it will reduce the amount of active principle present in the product. So that's what I just made it in very short about containers, closures. You have studied more in your pharmaceutics. You can add if they are asking essay question. And number 4, the frequency of testing. This is also we have discussed about that. Frequency of testing should be such that it is sufficient to establish the stability profile of new drug substance. Stability profile of new drug substance. For product with a proposed shelf life of at least 12 months, the testing frequency. So 12 months. So at a long-term storage condition, it should be every 3 months over the first year your test, and every 6 months over the second year, and annual therefore throughout the proposed shelf-life expiry date. So suppose if it is third year, you have to go for 3 years. Usually nowadays they made it in 2 years maximum. 3 years also available. Even 5 years also available. In the case of all accelerated storage condition, a minimum of 3 time points including the initial 0 and end point, the stability study. 0, 3 months, 6 months is recommended. Understand? And then when the testing is an intermediate storage condition, is necessary a result significant changes at the auxiliary storage condition. A minimum of 4 test points include the initial and final time points. It's recommended for 0, that means initial stage, and after 6 months, after 9 months, after 12 months. This already we have discussed in the previous slides. So this is how for the testing for intermediate storage we have to do. The next one is sample plan. How the sample should be taken. What is the sample plan? Already we know that there are various type of sampling is available. Whenever you are taking for any standardization or any type of evaluation, how the sampling should be done, we have already discussed in the first class itself. So here we made it into one slide, very simple. The sampling plan for stability testing involves planning for the number of samples to be charged or taken to this stability chambers and sampling out of charged batch. Which batch? So that over the entire study you are taking some sample which will relate to the overall batch, and the entire study will help for how you are taking randomly, how many numbers you have to take. These all they have given, every monograph they have given separately. Apart from that, in general, the first step should be the development of sampling time points followed by number of sample needed to draw for each pull out or each pull point for a complete evaluation of a test parameter, and finally add up to get the total number of samples. Total number of samples you have taken for a batch or a product. See here, for an example, there would be a requirement of about 100 tablets per pull out. You want to take 100 tablets according to the batch size. In a long term of accelerating stability studies, example, we are taking a tablets. So you are taking 10 tablets each for assay, hardness, moisture content. You are taking, and also assay also, you are doing assay and other tests also. But anyhow, here we have made only assay, hardness and moisture. You are taken 10 each, 30. It is 10 each means for assay 10, hardness tested for 10 and moisture content is for 10. So 30 it came. And 6 tablets for dissolution and for each dissolution and disintegration test. So 12 tablets you have taken. And 50 tablets you have to use for friability. So totally how much it comes? 36 plus 30 plus 12, 42 plus 50, that is coming around I think not 30 plus 12, it is 42 plus that is 92. So 8 tablets is again we can use for another test which can be done. So 50, 100 we have drawn. I made it only 3 tests in the that is why it is coming 92 and 8 tablets are still remaining when you take 100. But there is tests when you add 100, if there is 100. This is how for tablet we are told. So there is what are the parameters. So one formulation, there will be having different type of stability tests, different type of requirements by the quality control or quality assurance. So according to the sampling should be numbered, sampling to be taken. This multiplied by the total number of pull out, how many pull out you have taken, will give you the total number of tablets required for a study. So there is some calculation which will make you how many tablets you have taken. So same thing, how many samples you have taken in a cream, how many tablets, how many samples you have taken for a capsule, how many samples, how much samples have been taken for a raw material. So this all makes a sampling plan should be there. This is followed by development of a sample plan which include a selection of containers representing the batch as the whole, but it should be in an unbiased manner. Random, there should not be any bias. You are taking from the finished product, you are taking only in the last batch, or you are taking only from the down, or only from the top. So these you have to take in a random sampling and number should be specified. The last one is about the test parameters. You are concluding with this that the stability test protocol should define the test parameters that would be used for evaluation of stability samples. The test that monitor quality, purity, potency and identity, which could be expected to change upon storage, are chosen as stability test. Again I am reading that point. The test which monitors the quality, purity, potency and identity, very important point for any drugs, which could be expected to change upon the storage, are chosen as a stability test. Very important for a stability test. In general, test parameters when you want to write. We have already done, but it is common. Therefore, appearance, assay, degradation product, microbiological test, dissolution and testing, dissolution testing, moisture and are the standard tests performed for stability testing samples. So what are there? When you want to think about, when you want to know about, when you want to say about quality, purity, potency and identity of a product, it is necessary for you to go for assay, content of the constituents, content of the active marker, content of the active principle, assay it, appearance and degradation product and the microbiological test and dissolution and the moisture content and the standard test should be performed. Microbiological tests include what all the microbiological tests we can do, that means sterility testing, preservative efficacy and microbiological or microbial count as applicable for a product. And what type of microorganism, colonies, these all can be studied. There are many studies can be performed in microbiology assay. The batch used for the stability study must meet all the test requirements, including heavy metals, impurity of heavy metals, residue on igniting by ash values and residue by solvent extraction, etc. have to be done. Some of these are required to time of the product release, but not required to be repeated during the stability testing. Understand? Can you follow? This is what they have written, that is the time of the product release from towards the market. So before releasing, it is necessary for you to perform all these parameters. So that is the discussion end of the stability testing for herbal products and herbal preparations. So what I want to say is, I have given only certain points. In 3 classes I tried my maximum to give you some information, but there are many. I just made very streamlined. The handout which you have, the book which you are following may not have all these details. Kindly students, please make clear. Even I am not giving the complete details. I am just small details in my small knowledge which I have. But it's an ocean to go far inside this subject. So you just read, you just know the things. Some of the things which is not clear, please give me a comment. Please inform me freely, because if I done any mistake, please try to inform me so that I will correct myself. Thank you very much for being with me.